Yes. Delta G and delta G prime. Delta G0, then you're asking about what are we going to do? So these are the three terms that you will find, of which delta G is the actual difference of energy, actual free energy, free energy potential difference that decides whether a reaction will happen or not. So a reaction is decided always by the actual delta G. Now actual delta G you always have to measure, then you cannot really have a list of delta G values because it depends on the condition, it depends on the concentration of the reactants and products. And that's why books will always have a delta G0, which is the standard energy, and from that you will calculate your delta G depending on what concentration you have. So delta G will be delta G0 plus product over reactant. So this is the equation for calculating delta G that actually decides whether a reaction will happen or not. So this is the equation that you will find in any chemistry book. But in a biochemistry book, this will be delta G0 prime. And what is the difference? There will be no difference if no acid or base is involved in the product or reactant. But because biochemical reactions, many of them involve an acid or a base, that means you will have a H plus on the reactant side or the product side. So in those cases, the delta G0 and delta G0 prime will be different. The way it is different is that the pH of water, which is 7, that means the hydrogen ion concentration is 10 to the minus 7 molar. So if we take the log of that, that is incorporated into this delta G value. For any reaction that does not have a hydrogen, that doesn't affect, but if it has a proton as the reactant product, then they will be different. So because we are doing all reactions in water at neutral pH, it makes no sense to take the log of 10 to the minus 7 always and add it to the standard one. So that's why they added it already to all the standard free energy potentials. So what you will find is delta G0 prime in all the equations. And so this will decide whether a reaction will take place or not. And we saw examples where delta G0 prime may be negative for one reaction, but actually the opposite reaction happens in the cell. Why is that? Because that is decided by the products and reactants. So what is one example of that we did? Where the actual reaction happening is opposite of the standard free energy potential. Step 1, yes. Anything else? Any other classic example that I pointed out? There's another one that we will do in the citric acid cycle. But the one that is always pointed out in your books is creatine phosphate. It has a delta G0 prime of minus 43 kilojoule, whereas ATP is minus 32. So you would expect that creatine phosphate will transfer its phosphate to ADP to make ATP, which it does. But it does the opposite also, and that it will do when the concentrations of the two changes. So what will be the product when this transfers a phosphate? It becomes creatine. When this transfers a phosphate, it becomes ATP. So if concentration of ATP is high in the cell, then that ATP will be used to make creatine. If the concentration of ATP goes down, that means you don't have any ATP. So this delta G0 will be even lower. In that case, the creatine phosphate will transfer its phosphate to ADP to make ATP. So that's the function of creatine phosphate, to get you more ATP when you don't have enough in the muscles. Alright, so when you are doing exercise, all your ATP is used up, then this number 32 becomes lower because the product increases. Product ATP. When you hydrolyze ATP, it becomes ADP. So ATP concentration goes up, ATP goes down, and so the delta G changes. And now the creatine phosphate will transfer its phosphate to ATP. And when you are done with your exercise and there is aerobic respiration again, you are making a lot of ATP back again. So then that ATP will be used to transfer the phosphate to creatine, and then that will be stored in the muscles. So the reaction can go both directions, although the list of delta G0 prime will tell you that it goes in one direction only, because that is the standard free energy.
So which direction does it go in? Is it negative? In which direction does the standard energy talk about? Creatinine phosphate.
This is the equation. This is the equation, and if you have ATP giving ADP plus phosphate, when you have, so this is the product, this is the reactant. So when you are doing exercise, all your ATP is used up, and so the product concentration increases, reactant concentration decreases.
I mean, for creatinine phosphate specifically, the negative delta G there is talking about which.
That's the standard. The tables you get in your book will always be the standard free energy, that is this value. They cannot give you a delta G for every condition, so that's why they give you the standard. So those numbers don't mean anything if the reactant and products change a lot. They will mean if the concentration is more or less constant, but for ATP, as you know, depending on your situation, you may have more ATP or more ADP. So these numbers keep on changing. Anything else?
Explain the BPG.
What is the concept?
How does it increase in BPG?
The BPG is an inhibitor of oxygen binding, right? All the tools that we studied, BPG and proton, they inhibit binding of oxygen. And that means when you go to higher altitude, you are getting less oxygen in your lungs, and so you will be getting less oxygen in your tissues because you're starting with low, so you get low amount released. And so you want to increase the BPG, which lowers the binding of oxygen. In the lungs, the oxygen concentration is always high, so maximum amount will be bound to the oxygen. So here was the oxygen binding curve. Sigmoidal curve, right? Like this. So this will be low concentration, so somewhere here will be your tissues, somewhere here will be the lungs. Alright, so when the blood transfer goes from the lungs to the tissues, you are going to release, for example, this amount of oxygen. If you have a high altitude, then the amount of oxygen in the air is less, so the hemoglobin also binds less oxygen. And so let's say you have this much of oxygen bound. And so it will release you less oxygen. So from here to here it releases this much, which is not sufficient, and that's why you have breathing problem. But if the BPG amount increases, then the amount bound even at this oxygen concentration also increases. So the curve will be something like this with the higher BPG. And so now the amount of oxygen released is starting from here, the lower level, and this is also now at a lower level. So you are getting the same amount of oxygen because the binding of oxygen decreases due to the BPG. So as you go to higher altitude, the amount of BPG increases to lower the binding of oxygen.
Just like really quickly, so glycogen, does it have a reducing end or not?
Technically, no.
Because there's like a protein there, right?
There is supposed to be, if you are just studying the structure of glycogen in the beginning, you have shown one reducing end and many non-reducing end. But if you look at the synthesis of glycogen, for synthesizing the first bond between the glucose and glucose, the enzyme is not capable of doing that.
It's a primer.
So it needs a primer, and there is a protein called glycogen that acts as a primer and also as an enzyme. It is an enzyme that also acts as a primer, so it adds the first glucose onto itself. So what was supposed to be a reducing agent and actually doesn't exist, it is actually bonded to an amino acid of the enzyme. So depending on who is asking this question, how much depth is asked, so you can, the answer can be one reducing end and many non-reducing ends. But in reality, in the muscles and liver, there is no reducing end because there is a protein bound to that.
And is it dextrin? Limit dextrin is the form glycogen takes before the debranching enzyme.
That's right, yes. So when you remove all the glucose from the non-reducing end up to the fourth glucose from the branches, that product is the limit dextrin. And if we do the calculations, that will take out about half of the glucose. Because it is 10% branch, it's a mathematical thing. So there are 10% branches, and all the branches will be cut down to up to the fourth glucose unit, which means you can get about 50% of the glucose hydrolyzed from glycogen at a very fast rate by the glycogen phosphorylase, and then the rate slows down because the other enzyme has to remove the branches, and eventually you can get all the glucose out, but that will be a slower process. Yes.
Back to like the BPG, I just wanted to know why after 24 hours like finally like alleviates the breathing problem. Like why does that have to be after 24 hours? Is it just
Because the body takes time to make that BPG. So it takes about 24 hours. It doesn't really take to make the BPG, but it's an adjustment that you are making to first of all know how much BPG is needed, right? So it will be based on the need that it will adjust to that level. So that takes about 24 hours.
Makes sense.
Usually none of these pathways take 24 hours, but it is the process that takes 24 hours. It's not just the reaction. Reactions are always fast. Yes.
Can you go over a few cycle?
So if you have two opposite pathways, alright, so it looks like you people will not let me proceed today. So if you have two opposite pathways, you have to, so glucose, let's say you go to fructose 6-phosphate, and then
This is the cycle that gets nothing better.
fructose
This is the cycle that gets nothing better.
1,6-bisphosphate, and then onwards. So in the reverse direction, you need to go uphill in energy, and so that will be this phosphate will be hydrolyzed. So ATP will be released as ADP and phosphate, and in this direction you need an ATP. comes out as. So this is your investment of ATP that you made in glycolysis. In the reverse direction, you are hydrolyzing that. So this is a committed step. So this is the point where it will be regulated. And the regulation always happens for both the pathways. Both glycolysis and gluconeogenesis will be regulated simultaneously because it makes sense to turn off one if you want to turn on the other. What happens if you don't regulate that properly, then this pathway will be on and this pathway will also be on to a certain extent. And the net result that you are going from here to here and then from here to here, you're going in a cycle. And that is called the substrate cycle or a futile cycle. The net result of any cyclic pathway, there will always be some net result, and in this case it will be hydrolysis of ATP. So ATP is added and then phosphate is removed, so you are basically converting ATP to ATP and phosphate. So that means you are losing energy of ATP in the form of heat. And that's why it's called a futile cycle, because you're not achieving anything, but you are consuming energy in the form of ATP. So you don't want that, and that's why it is believed that all these substrate cycles are very tightly regulated. But not always. There are examples where they are not regulated on purpose. For example, in hibernating animals, they need the energy to be generated even when you are not doing any activity. And so they have these cycles turned on so that energy is generated this way. So this also leads some people to predict that this also happens in humans. There is no evidence of that. But there are some people who will say that they are doing everything but they are not gaining weight or losing weight, whatever you want to do. Whereas another person is doing that so efficiently. One of the reasons may be these cycles. These cycles may be operating in different people to different extent. So you may be losing energy or losing weight without effort, whereas another person is putting in more effort to lose weight. But whether that's true or not is, I don't know. This also brings, as a pharmacist, if you want to develop a drug, you want to develop something like this as a weight loss drug. You want to induce this to happen. And that way you can lose weight without doing activity. Yes?
The Cori cycle isn't the futile cycle?
No.
Why not?
The Cori cycle, yes. So the Cori cycle it's not a substrate cycle. It is happening in two different places, and the cell is not really thinking about making this a cycle. Right? You are, as an outsider, as a scientist, you are looking at it. Oh, this is the reaction happening here, and this is another reaction happening here, and the product actually travels from here to here, and that's why you are calling it a cycle. And that's what Cori did. These reactions happen in two different places. There are a series of reactions, it's not just one substance. So the net result is that glucose is converted to pyruvate in the muscles, and under anaerobic condition, the pyruvate is converted to lactate that goes to the liver, and then the liver is converted back to glucose. So you are starting with glucose and ending with glucose. The only difference is that you are using 4 more ATPs. You make 2 ATPs in glycolysis, you consume 6 ATPs in gluconeogenesis, so that means you are burning 4 ATPs in the process. And that's how you burn fat calories. When you are doing exercise, you are actually burning fat, not while you are doing exercise, but after your exercise, when all this goes up, and the pyruvate is converted back to glucose. Yes.
I just wanted to ask if you can go over the glycogen phosphorate, like the reciprocal regulation with insulin and glucagon.
Those are 2 separate examples of reciprocal regulation. right? So one will, again, there's no point in memorizing which one is activated, which one is inactivated. One will be activated, the other will be inactivated. And that happens by phosphorylation and dephosphorylation. Right, so insulin will activate a phosphatase, and that phosphatase will remove a phosphate from glycogen phosphorylase and will remove a phosphate from glycogen synthase, both. Because it's a phosphatase, it will remove a phosphate from both. And this act of removing a phosphate will activate one and inactivate the other. So you are always having one of them in active form. Either you're synthesizing glycogen or you're breaking down glycogen. You're not doing both at the same time. Because insulin, which removes the phosphate, and that act of removing a phosphate will activate one and inactivate the other. And in the same way, that's one example of reciprocal regulation. Glucagon is another example of regulation which also does the similar thing, it just does the opposite. So it will activate a kinase which puts a phosphate on glycogen synthase and also puts a phosphate on glycogen phosphorylase. And the result will be again the opposite. The act of putting a phosphate will activate one and will inactivate the other. So that is another example of reciprocal regulation. So what you are asking are two separate examples of reciprocal regulation. People always confuse, or students confuse, that glucagon and insulin are the reciprocal regulators. Now, they are separate reciprocal regulators. This is one and this is the other. Two separate examples. Number. What is the question? That's what I just answered. You were not here. So, what percentage of the carbon dioxide is transported and released in the lungs as bicarbonate and as carbamate? So it was 50% of the carbon dioxide that is exhaled in the lungs came as carbamate, reacting with the N-terminus of the hemoglobin, and the other 50% came in the form of bicarbonate dissolved in the blood. So it is half of the exhaled carbon dioxide from
biphosphate and bisphosphate?
Biphosphate term is not used, it's a diphosphate and bisphosphate. So diphosphate means the two phosphates are linked to each other, and bisphosphate means the phosphates are on two different positions of the molecule, like 2, 3 bisphosphates. So one phosphate at position 2 and one phosphate at position 3. Whereas adenosine diphosphate will be adenosine ribose, then phosphate, and that phosphate is attached to another phosphate. So the two phosphates are attached to each other. That is a diphosphate.
So I know that Alice works like the access regulation, but I just wanted to know like what would be the most common.
Allosteric regulation is the
Allosteric.
most common, yes, because there are more of them.
Oh. Okay. I want to know if during the exam we're gonna be like supplemented with scrap paper, because I like to like write my thoughts down.
Is that true?
Yeah, you get,
You get scrap paper?
usually, but that's up to you.
What do you want?
Piece of paper.
It's like anything.
Can you also word the answer choices in your wording?
Yeah, I can bring piece of paper, but there will be two groups I think kind of.
Yeah.
I don't want to make rules. Whatever the rule is, because it is allowed, then that is allowed.
Someone brings like that question about the delta G zero.
Yeah.
All of them were true, but
All true according to ATP, but not
according to why it's happening.
Yeah. Those are all true.
So,
I specifically for myself, so I didn't know
Because there is energy to that phosphate. you have energy of oxidation.
I didn't take it though.
The question is, this is an important question. None of the reactions we actually use free phosphate ion for any reaction. It's always ATP, GTP, so on. But this is one reaction in which actually a phosphate from solution is taken and a bond is formed to make ATP.
Which one?
So that happens in some reactions where you are doing an oxidation reaction. For example, this is an oxidation reaction using NAD, and that any oxidation reaction will always give you energy, and that energy is there, and that's why it forms a bond to the phosphate.
What reaction is that uses a free phosphate?
6.
The step 6, where you are using the glyceraldehyde 3-phosphate dehydrogenase with NAD, that oxidation by NAD releases enough energy to form a bond to free phosphate ions. These are one of the rare situations where you are actually forming a bond with free phosphate ion from solution. And this is where the extra phosphate comes from also. Another question, possible question can be if glycolysis gives you 2 ATP, gluconeogenesis takes 4 ATPs, where are the phosphates coming from? Right? It takes 4 ATPs, it takes more ATP. Where is the extra phosphate coming from?
In solution.
They are all coming from here. Because from the solution, from this particular reaction. It doesn't look like too many people in class. I didn't even give out the attendance sheet. But once again, as I said, the questions will be, I gave you the different topics like water, acid-base buffers, that's one topic. Amino acid, proteins and protein structure, that's another, and the peptide bonds, that's another, and the proteins, collagen, hemoglobin, that's another topic. Enzymes, coenzymes, mechanism, that's a really broad one. Metabolic pathways and high energy compound energetics, that's one, and then carbohydrate glycolysis, and other pathways which are gluconeogenesis, pentose phosphate pathway, and glycogen degradation. So that makes 7 different topics, of which probably the enzymes are a little bigger because they have everything included there. So if there are 7 different topics, there will be about 6 to 8 questions approximately each. Some of them more, some of them less. And there will be a total of 5 extra questions, not marked as a bonus question. There are just 5 extra questions. And why are there extra questions? Not because I'm a nice person. The extra questions are there because there will be no curving at the end. These are the extra, this is the opportunity that you get 4 extra questions. And that's why there are 5 extra questions. That means you can technically get 110 out of 100.
On the bonus easily.
But there's nothing marked as a bonus question. It's just in the picture.
So those 5 are technically the curve?
Yeah.
Alright, so 55 questions. 1 hour and 50 minutes.
You said 50 and 3.
Yeah, I know. He changed it.
All of them have a binding pocket where specificity is determined. So the serine, histidine, and the aspartame, those are at the catalytic site. And right below that will be the binding pocket that recognizes the substance. Because it won't cut any position part, for example, after lysine or arginine or trypsin, so that binding pocket will be a long one and have a negative charge at the end that recognizes it is part of the same enzyme. Enzyme is big, yes. It's not two different enzymes. It is the same enzyme, and the active site has a pocket where the side chain of the lysine fits.
So you said in class that basic amino acids act as acids at the binding site. Is that the same with histidine, or is it too neutral to act as an acid?
It does both. Histidine is the only one that can act as acid or base.
Okay, because we use like lysine and arginine as examples in class,
Of acids.
and then
Glutamate and aspartate as bases.
right, but we didn't talk about histidine.
Right. And histidine can be both. The histidine, in some enzymes, it is acting as an acid, some enzymes it is acting
Okay.
as a base. In fact, for the serine proteases, you saw there are two nitrogens of the histidine, and that helps to form the hydrogen bond. The catalytic triad is because of that, histidine having two nitrogens, so it can gain hydrogen from one side and lose hydrogen from the other side.
If everyone does bad on the exam, do you care what?
Professor, better.
No, you have the 5 extra points. Yes. What I say?
You think that's not gonna happen?
26 hours before exam. So I'll try to do it this whole thing is new to me. All these exams are things that just figuring out. It's not, the exam is not yet written, so it's not
Oh,
yet.
yeah. ChatGPT better go crazy, right?
Why does the deficiency of glucose mean dehydrogenase causing anemia?
Glucose 6-phosphate dehydrogenase, because that is the first step of the pentose phosphate pathway, and you make NADPH. There are 2 steps in which you make NADPH. NADPH is a reducing agent.
She's asking about why deficiency needs to be.
So this is your glucose 6-phosphate, and then you oxidize this,
Suicidalisis.
form the lactose. And you need an oxidizing agent. So this is the reaction. This is the first step of the pentose phosphate pathway, and the purpose of this pathway is to make 2 things. One is NADPH, which is made in this step, and if it is made in the next step also. Alright, so we haven't really, as we go along all these different pathways, you will find that there are too many pathways, too many reactions. We won't be doing all the reactions, I'll just point out the main ones. The ones that are more important, and every book has it, and every exam will ask you a question, are the glycolysis, gluconeogenesis, citric acid cycle, so these are the basic ones. Besides that, we will only talk about the important steps in other pathways. So the important step here is this one and the next one, where it is oxidized again, and it produces NADPH. And that is one of the important reasons for this pathway to make NADPH, which is a reducing agent and is used for mostly for fatty acid synthesis, which we will study later, because that needs a reducing agent. But the red blood cells especially needs NADPH because the red blood cells are in an aerobic environment, and so they easily get oxidized. Like the hemoglobin the iron can get oxidized, or there can be other reactions that happen with toxic side products from oxygen. So you need a reducing environment. What those oxidizing agents, like the peroxides, you need to reduce them, and one that reduces them is this glutathione. So glutathione also has this SH group, which gets oxidized. So 2 of them gives this disulfide, and this way the glutathione is used up, and you need to regenerate the glutathione, and that is done by the NADPH. So red blood cells have a special need for this pentose phosphate pathway, and that's why they have this pathway active all the time, because they need NADPH. And if you don't have the NADPH, then red blood cells will automatically lyse because the hemoglobin is destroyed, and the membranes are destroyed with binder peroxide. And so they need the NADPH, and this is an important enzyme, the glucose 6-phosphate dehydrogenase. So this is a common disease that some people have. Deficiency of this enzyme causes disease. Anything else?
Do you think you're going to reuse questions from the quiz?
What?
Do you think you're going to reuse questions from the 2 quizzes we have?
Sure.
Awesome, thank you.
Is it made already?
Is that the 5 curves?
Not all of them. It's not.
No, all 10 of them, please. There's 16 of them, at least you can go forward.
I think he knows that.
All right, so no one has any more questions?
Okay, what's the next?
Like, just to clarify for myself, questions aren't really a reflection of the
Maybe to see if like you study.
Everything will be related to, those are not questions, those are just points that I mentioned, right? Some of them are in the form of questions, some of them are just statements. So,
Some of them we didn't go over in class.
which one?
We'll just like, I don't know, some of them are just like, I can't think of a specific example, but some of them we didn't particularly.
I don't think so.
We did not cover Apple enzymes in class.
We went over everything that is in the book.
I don't think we talked about apo enzymes and hollow enzymes.
Someone asked me.
Okay.
We didn't talk about it in class.
No, not
We
no.
talked about it, but we didn't define these things. That's fine.
The define means if it is without the coenzyme, that's called an coenzyme.
Without a coenzyme, like a vitamin.
All right, so if no one has any questions, I don't think we have time for anything, so we will end it here. Yes.
Do we have to know about binding pockets in the serine proteases?
What do we know about it? It's there, yes. We already know about it. Yeah, you have to know what the binding, how they differ for chymotrypsin and
Whatever you're guessing.
Can you go over each one? Like for chymotrypsin, trypsin?
chymotrypsin. The trypsin has a recognition sequence that it cuts after lysine and arginine. So the trypsin will cut the peptide bond whenever it finds a lysine or arginine. There are some exceptions, and both these have a long chain, right? So you have the alpha carbon, and then there will be a long chain, and then you have a NH. So the binding pocket of this. So if this is your trypsin. So this is your trypsin, the active site, and this is where the reaction takes place. So this is your peptide bond starting from here to there, and the lysine comes and binds to this pocket. And the catalytic triad, which is the aspartate, histidine, and serine. So serine gives you a negative charge that will react at this carbon, and that's how you get the hydrolysis. So the catalysis happens here, the recognition of that substrate happens at this binding pocket. So that means the binding pocket must be long enough to accommodate this chain, and usually has a negative charge amino acid at this end, so that attracts the lysine. So that's the trypsin binding pocket. Chymotrypsin cuts after aromatic amino acid.
So I just understand.
means instead of lysine arginine, it will be tryptophan, alanine. Those have an aromatic group, so that means the binding pocket will be all hydrophobic. So there will not be any charge here. So you need to fit the aromatic group here. That means these will be hydrophobic amino acids that the end are not charged, not charged amino acids. So it's right size and
This is actually lysine,
right
lysine
charge
there,
or
milk lysine, that's how it like anchors down.
the other one is the elastase, which cuts up alanine. Alanine is a small amino acid, so the binding pocket.
Oh, it happens at the
So
top of
which
the
amino
chart.
acid is responsible for binding to the substrate? Would it be one of the like catalytic triads or
Binding pocket.
Catalytic triad doesn't bind, it catalyzes. Binding are the amino acids here in the pocket. So there will be amino acids, so this is one amino acid, this is one amino acid. So it's the length of the pocket that is important, and what is important is that there must be an aspartate or a glutamate at this end that traps the positive charge.
So the bond.
So we are talking about the amino acids forming the pocket, not
So
the
if it's a positive amino acid that it ends with, there's a negative electrostatic force. If it's a negative electro, if it's a negative
If it's
amino
CO
acid,
negative, for example.
and it's a positive electrostatic force grabbing.
So you just
All right,
which
so
amino acid
I
it
will
is.
see you on Wednesday.
Explain.
Thank you, Professor.
Thank you.